The various symptoms of HIV infection. We are aware that HIV is a collection of various symptoms of various diseases. So, lymph nodes, we are aware lymph and all the defense mechanism will be shut down, and as a result, the disease will start proliferating. So, lymph nodes they start swelling. So, there will be sudden outgrowths in the body at the places where there are lymph nodes. Respiratory system will collapse, and as a result of that, the individual will suffer from pneumonia, dry cough, as well as sore throat. Sore throat, it means there will be always itching i- sensation in the throat. Muscle pain will be there. The individual's joints will become weak, calcium deposition will be affected. You can think of arthritis to a heightened level, headache and difficulty in concentrating. Even the nervous system it starts deteriorating. The individual they have meningitis or primary stages of meningitis in this case. Skin, it shows rashes, fever is there, excessive sweating at night is also a symptom for the same disease. Digestive system is not working properly, and as a result diarrhea, vomiting and nausea, it means tendency to vomit is always there. The nails, they have curving, and at the corners there will be thickening for the nails. Loss of weight will be there. The individual will always feel tired, and these are some of the symptoms, primary symptoms of the HIV infection. We are not saying that this is symptoms of AIDS. Now, after this, the symptoms of AIDS will arise. The acute symptoms for the HIV infection is continuous fever, pharyngitis. There will be sores in the esophagus, muscle spasm will be there. There will be liver enlargement. Person will become very thin. Gastric problem will be there. Sometimes ulcer may arise. Cancerous lymph will be there. Lymph or cancer in the lymph will be generated, and the individual will have nervous breakdown. These are the acute symptoms. Now we can say that the individual is suffering from AIDS after this stage. There are various ways by which we can identify the virus, and one of the primary ways is we have already discussed about, and it is the ELISA. What is the full form of ELISA? ELISA is enzyme linked immunosorbent assay, and with this test we can diagnose the disease. The disease can be diagnosed in an HIV patient within 2 to 6 weeks of exposure of this disease. And once the individual is aware that he or she is suffering from the disease, they can do nothing about the disease other than waiting for the time period when the acuteness of the disease increases and they eventually die. There is no as such medication for the disease, and since it is a virus and it is an RNA virus, so the chances of of creating any vaccine is also a very big challenge for our doctors and scientists. What we can do, or like how can we detect the disease? Let's discuss it in a quick way. First thing is done in order to find out the disease is the blood serum of the patient is taken. At first blood is taken, then from the blood serum is segregated or separated out. This serum of the individual who is to be tested for virus infection is used to create a plate. Now, ELISA plates are certain microtiter wells. We have discussed in detail that this kind of chambers are there. 96 number of small wells are there. So each of the well will be given or will be supplied with some amount of serum, which is taken from the blood of the person who is to be tested. Now, from there, we will be adding this thing to the reaction tube. What is a reaction tube? What does it contain? It contains an HIV antigen which is coated on, or it is an inactivated HIV antigen which is taken. So blood serum will get combined, or blood serum from there, the combination of first antibody will be there to the antigen. Now, how will the antibody come? Think that the individual is actually a infected person. So the infected person will, once the person is infected, antibodies start developing in that individual. So this antibody, which is developed in an infected person, will come and bind with the antigen which is there in the reaction tube. And as a result, one antigen antibody combined substance will be formed. This antigen antibody combination will be as such that, or you can think that antigen will be absorbed, will be capable of absorbing the antibody of the same individual who is suffering from the disease. After that, the same antibody will be added with the antibody of second type, or we say that this is the antibody to HIV. So HIV antibody will add to this antibody which is already formed in the individual, and then the chromogenic substance, that is the enzyme, will be added, and as a result, if it is a positive test, then there will be color formation. If the color is seen, it means the individual is suffering from the disease. Otherwise, no specific color formation will be there. So what we are doing, we are just doing a series of steps. First thing we are doing is we are taking microtiter wells, we are adding the blood serum with the antigen. So antigen is getting added with the serum of the individual who is already suffering from the disease. This serum will be containing the antibodies as well, because the individual is already exposed to the antigen of HIV. Now, when this combines with the antibody, then after this, antibody 1 will be added to the HIV individual's antibody. So this will be a HIV antibody, and finally the color will be developed with the addition of enzyme, and which is a chromogenic substance. So this is a simple way by which, just by detection of the color, we can diagnose that a person is suffering from the disease or not. Now, in order to confirm this, we do Western blotting. Western blot is used in order to detect, or in order to find out the HIV protein. HIV is a protein. Whenever we want to detect a particular kind of protein, we go for Western blotting. The similar way Southern blotting is there, we have already discussed about it. So, anyways, how is it done? Gel electrophoresis will be there, and nitrocellulose membrane will be used in order to transfer the antigen over the nitrocellulose membrane, and it will be incubated for a certain period of time, and if there is antigen antibody reaction taking place, then there will be detection of color, and as a whole, this result will give us an idea that is the individual HIV positive or not. So just in brief, you have to remember this process. Now, the entire action or the working mechanism of the RNA virus or retrovirus, HIV retrovirus. Whenever any individual is encountering a viral infection, at that time it is very important that we understand that the viral infections, whenever anyone suffers from it, it is as such that we cannot expect that we can find a vaccine or a recovery way to the RNA viruses. The reason for that is we are aware about the stability of the DNA and RNA. DNA viruses, they mutate very slow, or within a course of 100 or 200 years they will be mutating. But the RNA viruses, they are prone to mutation at a very, very rapid speed, and because of this rapid speed of mutation, we find it very difficult to think about or to form an antibody for this disease, because by the time we form an antibody, the virus has already replicated and formed a completely new virus. So this makes the vaccine formation quite difficult for most of the RNA viruses. We are going to discuss in details about how the infection is going to increase in the RNA viruses. But when we discuss about it, few things are to be kept in mind. That RNA virus, when it is getting into any individual, it is at first getting uh, converted into, it is converting the genetic material from RNA to DNA with the help of reverse transcriptase. That basic idea should be there. Now let's begin off with the discussion. First thing which happens during this time is an HIV virus. For the structure of HIV virus is quite important. It contains glycoprotein outer coat, two RNA strands, and these both single RNA strands have presence of reverse transcriptase enzyme. Envelope is also there. Now, as soon as the RNA virus attacks any human cell, at that time it just goes and combines with the, or gets attached to the individual's body, and then after it starts pushing its genetic material inside the individual or inside the cell of the infected person. As soon as the virus has injected, first thing is fusion of the HIV to the host cell surface. So here fusion doesn't mean that the entire virus is getting inside. We are aware viruses only inject their genetic material. So the both the RNA strands will be injected. That is what is happening in the second case. HIV RNA reverse transcriptase integrase as well as the viral proteins enter the host cell, not the outer coat. After that, the virus, it first thing it is going to do is use the reverse transcriptase enzyme and convert its genetic material from RNA to DNA so that it can now enter the living system or the nucleus of the cell to which it is exposed to. So viral DNA is formed by reverse transcriptase. That is the third step. Fourth step is that the viral DNA is transported across the nucleus and integrates into the host DNA. How will it induce itself inside the DNA of the host? By using the exonuclease activity, and with the help of exonuclease it will cut both the ends of the, or just cut the end of a DNA where it can get incorporated, where the nitrogenous bases can fix. And then after, once it has got inside the system of the RNA, or has got inside the human's nucleus, then the entire machinery of the host cell is hijacked. Once the hijacking has taken place in a complete way, after that we can just see that only the host protein as well as the entire cellular machinery will be used in order to form the viral protein. Only the viral protein will be formed, outer coat, the glycoprotein, as well as the RNA will be formed. And when the entire cell has formed enough amount of protein as well as the coat, protein coat as well as the RNAs, now the combination or assembly of the entire virus will take place. The complete feasible or living virus will be formed, and at the end the entire cell will be burst open in order to release all the viruses in the nearby cells. So the infection will go on increasing. At this stage, when the virus is going to burst the cell off and release its similar kind, at that time the same virus is known as a viron. The fifth step we have discussed. Now the fifth step is new viral RNA is used as genomic RNA, and it is used to make the viral proteins. The new viral RNA and proteins move to the cell surface and a new immature HIV forms. So at the seventh stage, we can see that the virus is released, viral proteases cleaves new polyproteins to create mature infectious virus, and this infectious virus will cause disease or will be infecting everyone, every other cell of the body. As a result, the individual will be capable of expression of the disease, the symptoms of the disease. How can we prevent the disease? We can prevent the disease by avoiding all the causes of the disease. That is, we should avoid sexual contact with unaware or If we are not aware that if the individual is suffering from the disease or not, we should avoid sexual contact with that person. Infected needles should be avoided. We should check that every time a doctor is giving you injection, at that time the doctor is using a new needle. Make sure that the syringe is teared off in front of you. Third thing is avoiding blood transfusion with and checking the blood transfusion, that is it from a infected HIV infected person or not. And even help while helping any person who is meeting with any accident, or who has um, who needs help at that time, making it quite evident that your bruise or your injured area is not exposed to the blood of the same, of another infected individual or any unknown individual. And the females who are expecting to get pregnant, those females, they should always get the HIV test done. That if the female is already suffering from the disease, then she should always get aborted, because the life of the child who is going to come to the world will be very troublesome. So these are few of the ways by which we can prevent the disease. Now, why we are not discussing about the vaccination or any other way out? Because we don't know till now that is there any vaccine possible or not. Still we are trying to create some vaccine. We can hope, we can be hopeful that they will come, there will be a time when the vaccine will be formed. But till then, these are few of the ways by which we can avoid the disease.